Hello, my name is Olivier Jarry and I'm CEO of Libera Bio. Our goal is to deliver large molecules, biologics, to intracellular targets in cancer. Libera Bio benefits from 3 decades of work of Professor Maria José Alonso. She is teaching pharmacology at the University of Santiago de Compostela in Spain. She also worked in Paris Sud and at MIT with Bob Langer. She is recognized in the area of drug delivery. She was past president of the Control Release Society and has received numerous awards, such as one of the most influential researcher last year, alongside Jennifer Doudna of CRISPR fame. She has published numerous articles, and her patents related to cancer are exclusively attributed to Libera Bio. Our team also includes Dr. Terrejo, who is managing our operations. She has a combined experience in academia as well as biotech. Myself, Olivier Jarry, coming from large pharma, Novartis, Bayer, and Bristol Myers Squibb, but also biotech in the last 10 years. Our advisory board includes Professor Langer from the MIT and a range of oncologists and nanomedicine specialists, especially Dr. Vicent and Dr. Macarulla, who are leading laboratories in Spain, in Navarra and in Catalonia, that are participating in our experiments and confirming our findings. We've been incubated at the University of Santiago. We received support so far from the region of Galicia, also Spain and the European Union. And we are now in a Series A after raising our seed money to humanize our lead candidates. We've received validations from a number of entities that you see here, and also awards, one with Johnson & Johnson, Quickfire, 1 year ago, and also actually 2 from Aim High and a few others, regional, national, and global. What are we trying to solve? We are trying to improve the standard of care for cancer treatment. Small molecules are typically highly toxic and they engage only about 10% of all targets. Monoclonal antibodies used in the last 10 years are much more specific and strongly engage their targets, but they are too large to penetrate by themselves the cancer cells and reach the intracellular targets. So our technology is aiming at bringing actually these antibodies to these intracellular targets. It consists of nanocapsules that are enveloped by polymers and can be functionalized in different ways. Hence the name we give them of Multifunctional Polymeric Nanocapsules, or MPN, and our MPN technology. These capsules are flexible, but at the same time they have a shell around them that protects them, for example, in plasma or against macrophages. So they are not only drops of lipids that wouldn't be protected, they are not PLGA nanoparticles that would be rigid. They are a hybrid that has very interesting properties. First of all, in the oily core, we can place a small molecule, and we have experimented more than 10 small molecules, for example, docetaxel. And this oily core is surrounded by a surfactant shell and then one or more polymer shells. When we deliver an antibody or another biologic, we place this agent between the shells on the periphery of a nanocapsule, and they are not protruding to the outside. So essentially they are hidden and protected once they are delivered or during their delivery by the nanocapsule. We can functionalize the outer layer by fixing a targeting ligand. This targeting ligand is aiming at cancer cells and then facilitates the penetration of the nanocapsule inside the cancer cell. We use LIP1 or a fragment of LIP1 that has interesting properties of both targeting NRP1 and NRP2 receptors on the surface of a cancer cell, bringing the nanocapsule closer to the surface, but it is also a cell-penetrating peptide that helps bring the entire nanocapsule inside the cancer cell. As said, we tested on 10 small molecules and 6 antibodies. Our lead candidates are KRAS, and we'll see them a little bit later. Our ingredients for the nanocapsule itself are all FDA and EMA approved, simplifying the processes for toxicology and approval. And the manufacture involves different processes for small molecules or biologics that are all very simple and scalable. We've done batch manufacturing, we've done continuous flow manufacturing, and we know that our nanocapsules can also be freeze-dried. Let's start with facts and the biodistribution of the nanocapsules. You see here the example of docetaxel, a small molecule that we have put in our nanocapsules MPN, and the high bar in the center of the chart represent concentration in tumors. You see the green bar, which is the advanced version of our nanocapsule, is about 40 times the height of the blue bar in this tumor, which is docetaxel injected directly, about 40 times. On the right side, you see different lymph nodes, and we see activity of a compound against metastases. On the left side, you see different organs and not a specific concentration, for example, in the liver, where they may be eliminated. So we believe that this allows us to deliver small molecules in an efficient way, very targeted, so you can get better safety and efficacy. For example, we may reduce the dose that is injected, but importantly, the nanocapsule and the active ingredients go to the tumor where they are going to be effective. Now let's move to antibodies. You see here one antibody, anti-gasdermin, that we tested and generated evidence with fluorescence. On the right side, you see over a sequence of pictures, actually of photographies, the endosomal capture, and the endosome is red, the antibody is green, and we see a co-location, a superposition of these two in the early hours after intravenous injection, and then they separate, giving us an indication that the antibody has been liberated from a nanocapsule and is now navigating inside the cytosol. You have here an overview in vivo of the same phenomenon, and on the left side a chart indicating stability in plasma. Even more interestingly, you see here some pictures taken with our anti-KRAS. On the right side, you see that the green dots have migrated to the inner side of the cell membrane where the KRAS is located. So this indicates that not only the nanocapsules have entered the cytosol, they have been submitted to endosomal capture and release, but the antibodies themselves have been liberated and have migrated to their KRAS target on the inner side of the membrane. Why KRAS? We are working mostly with G12D and G12V at this point. Each of them is present in about a quarter of pancreatic cancers, about 1 out of 10 colorectal, and 1 out of 20 lung cancers. So very interesting targets, knowing that we can deliver a broad range of antibodies addressing many other targets. In practice now with in vivo models, you see here two of these models. On the left side, a lung cancer model where our MPN anti-KRAS has greatly reduced the growth of a tumor in this lung cancer model. On the right side, a colorectal cancer model. Interestingly, at the bottom right, you see this necrosis of the tumor itself, 71% and a bit later 83%, showing that maybe the tumor has not regressed yet, but it's largely necrosed inside. Going to pancreas, you have here two more models. On the left side, we have a xenograft model where we measured the expression of phosphor-ERK minus 30% and the apoptosis increased by 9 times, and a similar reduction of the growth of a tumor after a number of days. The right side is particularly interesting. This is a PDX pancreatic model that is very demanding. Our oncology team proposed to do this experiment to show if there was really a signal with our product, knowing that the standard of care, gemcitabine and Abraxane, is not very effective. And you see here the middle curve in gray is actually very close to the standard. When combined with our product in green, at the bottom, you see that essentially the growth of the tumor is stopped and not progressing. We have a range of patents around the world to protect the nanocapsule, the delivery of antibodies, and the KRAS construct. We have long explanations how this is superior to the other attempts to deliver antibodies. Ours are not engineered. They are the whole antibody. We can also deliver fragments, but essentially manufacturing is simpler, and we believe there is a more natural affinity to their targets. Our goal is to develop these two lead candidates, to collaborate possibly with them, but to collaborate also on new antibodies with pharma partners, and at some point to also license the small molecules or the technology for small molecules. The technology greatly improves the delivery of small molecules, and we believe that a number of companies could benefit from it. We have had two partnerships, contemplate another one to deliver cytotoxic that would better be targeted to the cancer cells. Where we are is we are in the middle now of humanization and characterization of our MPN with the two lead candidates, anti-KRAS. We are going to select one and probably towards the middle of 2022 enter enabling studies, CMC, in order to seek for an IND, IMPD approval towards the end of 2023. And we are starting to raise the amounts needed here, $8.4 million, 7 million euros, to conduct these enabling studies. In summary, we have a completely patented and exclusive technology to deliver biologics such as monoclonal antibodies, but other biologics such as siRNA, to intracellular targets. We show evidence of both internalization and tumor shrinkage. Manufacturing is simple and scalable. Our team is very experienced and dedicated to develop this technology and make it available to others. We are currently starting our Series A in order to lead the enabling studies and CMC, and we are very open to global alliances and out-licensing the technology. Feel free to contact me. The contacts are here on this page, especially contact@liberabio.com. Thank you.